Good morning, my dear students. In quarter one, we are going to study the prokaryotes. What are prokaryotes? Prokaryotes are living microorganisms surrounding us nearly in all the environments. Mainly, prokaryotes is derived from a Greek word which means before nucleus. Prokaryotes are divided into two domains, bacteria and archaea. Prokaryotes live everywhere surrounding us, like we said before, but in extreme environments, not all the kinds can survive. Unlike archaea, bacteria can survive in extreme environments, but archaea can stand the hostile environments. Okay, so we can call archaea extremophiles because they can survive in extreme conditions. According to the environment, the extreme conditions surrounding the archaea environment, we can classify archaea into three main groups: thermoacidophils, halophils, and methanogens. We are going to talk about each group separately. So the first group, we called it thermoacidophils. From the name, thermo mean it can survive in high temperature, okay, and acid, they can live in acidic environment. The part of the word phils mean that like. So thermoacidophils mean that this kind of archaea likes high temperature and acidic environment. This kind of archaea can survive at high temperature above 80 degrees and in acidic environment very low. The pH of this acidic environment can reach 1 or 2. Example for the environment where archaea or thermoacidophils can exist is hot sulfur springs. Okay, about their method for respiration, mainly thermoacidophils are anaerobes. What is meant by anaerobes? Anaerobes means that they can survive in presence of oxygen. If oxygen is present, this kind, thermoacidophils, will die. The second group is called halophils. Halophils. We said phils means like, so halo means salt. They like high salty concentration. Like where? Like the Great Salt Lake. So halophils like high salt concentration, so they can survive in places such as the Great Salt Lake. For their method of respiration, they are aerobes, unlike thermoacidophils. Halophils are aerobes, which means, right? It means that they need oxygen. Oxygen is essential for their survival. In this kind of archaea, they can act like plants. They can carry out photosynthesis, but there is no chlorophyll inside these halophils. They use another thing. What is this? It's a certain kind of protein which allow these kind of archaea to perform photosynthesis. The third group is called methanogens. Methanogens. Methanogens means they can produce methane gas. Okay? Gens means to create or to produce. Methane is derived from, of course, the methane gas. So this kind of archaea can be found in sewage treatment plants, in swamps, and in volcanic vents. Okay, from these places, you can figure that this kind can tolerate very high temperature. Okay, okay. We said that thermoacidophils are anaerobes. They can survive in presence of oxygen. Halophils are aerobes. So methanogens are like thermoacidophils. They are anaerobes. So like thermoacidophils, they can survive in presence of oxygen. And instead, they use CO2, carbon dioxide gas, in respiration and produce methane as a waste product. That's why they are called methanogens. Now, we are going to discuss the structure of the prokaryotes, bacteria and archaea. This is the prokaryotic cell. It's somehow different from the eukaryotic cell. They have DNA and ribosomes inside, like eukaryotic cells, but they lack some other organelles, okay, like the nuclear membrane and other organelles present in the eukaryotic cell, like mitochondria, okay, which is present in the eukaryotic cell. It's responsible for creating energy, producing energy. These kind of prokaryotes lack these organelles. So here's the structure of the prokaryotic cell. From the very outside, you will find the capsule. Okay, capsule is mainly for protecting the cell from other organisms and to protect it from drying out. Beneath the capsule, this red line, this is the cell wall. Okay, it also protects the cell and it's important to give the cell its shape. Under the cell wall, there is the cell membrane. The cell membrane regulates the type of molecules that move into and out of the cell. Inside here, you will find the cytoplasm, which contain other components of the cell, like DNA, ribosomes, and any other organic compounds that the bacteria or archaea use for living. This is the chromosomes. Okay, chromosome carry the genetic information from one generation of the bacteria or archaea to the other generation. These parts, these extensions are known as VIS. VIS is very important for the prokaryotic cell because this VIS help the cell to attach to the surface of the environment surrounding it and to attach to other bacteria or archaea during what's called conjugation. We'll discuss it in a minute. These long filaments are known as flagellum. It's present in most of prokaryotic cells and it's important to help the prokaryotic cell to move. So flagellum is important for motion. Capsule, it's a layer surrounding the outer part of the cell. It's made of something called polysaccharides. Polysaccharides means sugars, molecules connected to each other, making a polysaccharide. Why capsule is important? It is important because it prevents drying out of the cell. It helps the cell to attach to other surfaces and protect the cell from being engulfed by white blood cells inside the blood of other organisms. Chromosomes, like we said, carry the genetic characters of the prokaryotic cell. Pili, or the singular is pilus. This is the projections that we talked about, and we said it's important to make like a bridge between two prokaryotic cells to allow conjugation. Conjugation is mainly to help transfer the genetic material. Next session, we are going to discuss the characteristics of bacteria and their way to replicate.